Research is the cornerstone of innovation in clinical practice, driving improvements in patient outcomes and shaping the future of medicine. Welcome to the Global Rhinology Network Topical Research Symposium. I'm Ashok Rokde from Winchester, UK, and it is my privilege to welcome you as a host. Joining me as co-hosts are Louise Melia from Bath, UK, and Professor Thibaut Van Zelle from Ghent, Belgium. This symposium showcases fascinating topics across 4 key modules: General Rhinology, COVID session, and Research in Chronic Rhinosinusitis. A special highlight is the PhD session featuring research conducted by enthusiastic young rhinologists who represent the future of our field. This symposium brings together leading global minds in rhinology who will highlight latest developments with a focus on translating research into impactful patient care. The Global Rhinology Network is dedicated to advancing knowledge innovation. Thank you for being part of this exciting journey. Thanks to Medtronic and Karl Storz for their generous support in making this symposium possible. Welcome to GRN-Topical Research Symposium.
Our next speaker will be Wout Backaert, also from the KUL Leuven in Belgium, and he will speak about hyperreactivity in upper airway inflammation.
Hello, good evening, everyone. I'm going to try to share my screen. It should be visible right now. Is that correct?
Yes.
All right, thank you. Thank you also to the organizers for giving me the opportunity to present part of my PhD here on this Global Rhinology Network research session. I am just looking to get a laser pointer. I recently finished my PhD in September this year, and currently I'm continuing my clinical training. In my PhD, I focused mainly on nasal hyperactivity in rhinitis and rhinosinusitis. Now, all of you probably know that about 6 symptoms are the cardinal symptoms of chronic upper airway inflammation. And depending on the way a patient presents with these symptoms and depending on the clinical tests, we can categorize them often in rhinitis and rhinosinusitis phenotypes. Now, patients with chronic upper airway inflammation, they can have these symptoms persistently or chronically, or for example, upon encounter with the specific allergen they are sensitized to. But these symptoms can also be triggered by specific environmental stimuli, such as temperature changes, humidity changes, air conditioning also, smoke, and so on. And this is what we call nasal hyperreactivity. Now, nasal hyperreactivity can be diagnosed, roughly speaking, in 2 different ways. We can diagnose it subjectively by simply asking the patient 2 questions. First, we ask the patient whether or not he or she has nasal symptoms that are triggered or exacerbated by specific environmental triggers, such as temperature changes and humidity changes. And in this case, we ask a second question, namely that if they have these symptoms, do they last for longer than 10 minutes? This is to really differentiate from the true hyperactivity from the normal physiological responses that take place in many normal and healthy individuals. On the other hand, hyperactivity can also be diagnosed objectively by means of a cold dry air provocation test. During this provocation, patients are exposed to air of -10 degrees for 15 minutes. And before and after the provocation, we measure their peak nasal inspiratory flow, which is kind of a measure for the nasal patency of the patients, and hence is also a measure for the nasal obstruction. And patients are considered to suffer from objective hyperreactivity if they have a decrease in peak nasal inspiratory flow of 20% or more. the first thing I wanted to do in my PhD was actually try to map out what the prevalence would be of nasal hyperactivity and to what degree these patients would be suffering from hyperactivity. So therefore, I performed a questionnaire study on more than 150 healthy control subjects and more than 600 patients with any kind of chronic upper airway inflammation. And we could see that nasal hyperactivity is not just limited to patients with non-allergic rhinitis, where it is mostly studied, but actually all phenotypes of upper airway inflammation have some patients that suffer from this nasal hyperactivity without differences in between these groups. So hyperactivity is really a common feature of any kind of chronic upper airway inflammation. Now, this figure schematically represents the innervation of the nasal and paranasal mucosa. And what's actually taking place is that these sensory neurons, these trigeminal sensory neurons, they express strip channels. Strip channels are nociceptors, they can sense the environment, and in normal physiological conditions, the strip channels can be activated. Then a signal is transmitted to the central nervous system, which usually responds with an efferent parasympathetic response, leading to vasodilation and increased mucus secretion, which leads to nasal symptoms like nasal obstruction and rhinorrhea. But nasal hyperactivity is mostly studied in patients with non-allergic rhinitis. And there it has been observed that these patients, they have a stronger innervation of the nasal mucosa, they express higher levels of this nociceptor here, TRPV1, and also have higher levels of neuropeptides such as substance P in the nasal secretions. So it was postulated that nasal hyperactivity could be the result of sensitized sensory neurons. And then these sensory neurons are strongly activated upon any encounter with any kind of trigger in the environment, which can then directly lead to release of neuropeptides from the sensory neuron itself, which can cause vasodilation and increased mucus secretion, leading to nasal symptoms. But of course, we could see that nasal hyperactivity was not just limited to patients with non-allergic rhinitis. And for example, it's also present in patients with allergic rhinitis or chronic rhinosinusitis with polyps, which are usually considered to be more Th2 mediated. So we want to investigate what the pathophysiology would be of nasal hyperactivity in these type 2 mediated upper airway diseases. To study this, we set up a study with 18 healthy control subjects and 45 patients with allergic rhinitis or CRS with nasal polyps. We invited the patients to come to the hospital twice. First, we took some baseline measurements, some baseline biopsies from the nose itself. And after the biopsy site had time to heal for 3 weeks, patients came back for a cold air provocation test. Before and after the cold air provocation test, we took some nasal secretions. We also took nasal tissue, as I already mentioned, and we measured the peak nasal inspiratory flow to get an idea of the reactivity of the patients to the cold air provocation itself. Now, as you recall from my introduction, hyperactivity can be diagnosed subjectively and objectively, but in both cases, it's usually seen as an on or off phenomenon. But we were thinking that it's probably unlikely that this approach satisfactorily reflects what's actually biologically going on in the sinonasal mucosa itself. So therefore, we included 2 other parameters, 2 continuous parameters. For the subjective parameter, we just asked the patient to indicate on a VAS score to what degree he or she suffered from exacerbated symptoms due to the cold air provocation. And for the objective parameter, we looked at the change in peak nasal inspiratory flow, the change in nasal patency as a continuous parameter, rather than working with this cutoff of a decrease of 20% or more. And on this figure here on the bottom of my slide, I indicate on the y-axis the change in peak nasal inspiratory flow, the change in nasal patency. And you can clearly see that even though not all patients with subjective hyperactivity, they do not all reach the cutoff of minus 20% decrease in peak nasal inspiratory flow for objective diagnosis of nasal hyperactivity, but we can clearly see that they have a strong decrease when you compare them with healthy control subjects or with patients without subjective nasal hyperactivity. So that's one thing. But of course, we're also wondering what would be, which factors could be predisposing for the reactivity to cold air provocation. So in the nasal secretions of these patients, we also measured several different neuropeptides, but also cytokines and so on. And here on the y-axis, again, you can see the reactivity to cold air measured by the change in peak nasal inspiratory flow. And we can see that for all these neuropeptides, substance P, neurokinin A, calcitonin gene-related peptide, for all these neuropeptides, there is a correlation between the levels of the neuropeptides at baseline and the objective reactivity to cold air provocation. And what's also very interesting, when we look in the tissue itself, we can see that in the tissue, there's also a correlation when you look at the expression levels of these TRP channels, TRPV1, TRPM8, but also of the pan-neuronal marker PGP9.5 and the change in peak nasal inspiratory flow. So the higher the levels of these neurogenic markers and these neuropeptides, the stronger the reactivity of the patient would be to the cold air provocation afterwards. What's also very intriguing, we could see a similar correlation when we looked at histamine. While histamine is also, or is usually considered to be more linked to type 2 inflammation. To study this a bit further, we cultured trigeminal neurons from just normal, healthy wild-type mice, and we exposed these neurons to capsaicin, which is a specific and very strong TRPV1 activator. In between the two applications, we administered Krebs solution for control or histamine to see whether or not histamine could have an effect on the sensitivity of the neurons for capsaicin at very low doses. And indeed, we could see that if the neurons received or were exposed to histamine in between the two applications, a higher number of neurons was actually responding to the second application when you compare it with the first application so there is a sensitization of the neuron by histamine during the interval between the two measurements. And this sensitization is probably mediated via an H1 mediated pathway, because when we apply pyrilamine, a specific H1 antihistamine to the culture, this effect is completely abolished. And what's more, these results were obtained for TRPV1, as I show you here, but also similar results were obtained when we challenged the neurons with cinnamaldehyde, which is a specific TRPA1 agonist. So we all now know that histamine can maybe also play a role in hyperactivity of the upper airways. Now, lastly, we were wondering what would be actually taking place in mucosa of these patients during the cold air provocation itself. Now, we actually failed to find evidence of increased levels of neuropeptides, substance P, neurokinin A, calcium gene-related peptides, when you compare the after cold air provocation measurements with the pre-provocation measurements. On the other hand, when we look at the epithelial cytokine IL-33, we can clearly see that this has a strong increase during the cold air provocation. So cold air provocation itself leads to activation of leads to epithelial activation. Now, to summarize these last results in one slide, I made this this figure. On the top, you can see that nasal hyperactivity is not, we cannot really speak about hyperactivity. It's more part of a continuous spectrum ranging from physiologic to real complete hyperactivity. And the stronger the reactivity is of the patients to the cold air provocation, the higher the levels of neuropeptides and histamine, the higher the TRP channel expression and the expression of PGP9.5. And I also showed that histamine is usually considered to be a type 2 mediator, can also sensitize the TRP channels present on these sensory neurons for their specific agonists. Now, of course, I did not do all this work just purely by myself. There was a lot of input from several professors, but also my PhD students helped me a lot with various experiments. So I want to thank them for all their help. But if you still have any questions, I'm really happy to answer them.